Hi CSBR members, the title of the journal that I'm going to introduce today is A Versatile Microbial Platform as a Tunable Whole-Cell Chemical Sensor, which published on Nature Communications on September 2024. The reason I chose this paper is that I've recently been focusing on the PYP-based whole-cell biosensor and found this paper can help me design, build, and test a whole-cell biosensor. Here are the highlights of this paper. In this study, the authors engineered Pseudomonas putida for whole-cell sensing of a variety of chemicals to overcome the typical limitations of biosensors. Need for transcription factors engineered to recognize specific molecules. They also integrated fluorescent reporters with synthetic orthotrophies with central metabolism that can be complemented by target analytes and growth-coupled setups. The platform enables the detection of a wide array of structurally diverse chemicals under various conditions, including co-cultures of producer cell factories and the sensor strains. The applicability of this versatile biosensor platform for monitoring complex biochemical processes, including plastic degradation by either purified hydrolytic enzymes or engineered bacteria. Therefore, the microbiome system in this study provides a rapid, sensitive, and readily adaptable tool for monitoring cell factory performance and for environmental analysis. Whole-cell biosensors are living microbes that convert the presence of a metabolite into a simple readout, such as growth or fluorescence. Such whole-cell biosensors are very useful for screening cell factories because they can directly sense extracellular products in supernatant, in vitro enzyme reactions, or co-cultures. However, classical biosensors usually rely on specific transcription factors, which must be redesigned for each new target and limit throughput. Therefore, authors replaced exploiting native assimilation pathways and coupled growth to fluorescence, creating a more general growth-based whole-cell biosensor platform. In this study, the authors engineered Pseudomonas putida into a whole-cell biosensor platform by wiring target chemicals into central metabolism so that cell growth and the constitutive fluorescence directly report analyte availability. Pseudomonas putida's broad native capacity to use aromatics, volatile fatty acids, sugars, and organic acids provides many metabolic entry points, while synthetic orthotrophies can be created by blocking specific carbon routes and complementing them only with the analyte of interest. In this growth-coupled design, biomass formation becomes proportional to the concentration of the essential molecule, and a chromosomally integrated MSF-GFP reporter converts growth differences into a quantitative, sensitive fluorescence signal over a wide concentration range. To enable growth-coupled detection of chemicals in sugar-based bioprocesses, the authors first blocked glucose use in Pseudomonas putida. They introduced 10 inframe deletions targeting glucose uptake, periplasmic oxidation, and downstream catabolism, thereby disabling utilization of all three native hexose substrates, glucose gluconate and 2-ketogluconate. In parallel, they chromosomally integrated a constitutively expressed MSF-GFP under the P14G promoter via TN7, so growth could be monitored by fluorescence. The resulting strain, Pseudomonas putida sens, served as the base chassis for all later biosensors and, unlike KT2440, could not grow on any hexose but grew well on glycerol, fructose, TCA intermediates, pyruvate, and PCA. On these alternative substrates, biomass and fluorescence were linearly correlated, validating MSF-GFP as a quantitative growth readout for chemical sensing. In their first application, the authors used the base sense strain to sense PCA produced by another Pseudomonas strains. Sense cannot grow on glucose but grows well on PCA, so GFP per OD increases linearly with initial PCA concentration over 3 orders of magnitude, with growth dropping about 50 millimole due to toxicity, which defines the practical upper limit. In cell culture, a Pseudomonas putida PCA producer grows on glucose and secretes PCA. Sense can only grow by consuming the secret PCA, giving over threefold higher GFP than the producer alone, and clearly above controls, including wild-type Pseudomonas putida. Thus, the platform race metabolites directly in co-culture without engineering the producer and offer a wide, near-linear dynamic range, superior to typical logarithmic transcription factor-based BCA senses for screening high-titer producers. In their second application, the authors adapt the sense platform to detect D-lactate secreted by E. coli producers. They start from Pseudomonas putida sense and build a new strain sense L by deleting ACA to prevent growth on acetate overflow from E. coli, and TSSA1 to inactivate the type 4 secretion system, making co-culture compatible. In monoculture calibration, sense L shows a nearly linear increase in MSFGFP fluorescence with increasing D-lactate over roughly 0.15 to 20 millimole. So fluorescence per OD directly reports extracellular D-lactate levels. To demonstrate utility, they construct an E. coli DLD knockout strain carrying a promoter RBS library driving Pseudomonas putida LDHA, generating variants with different D-lactate productivities. Supernatants from these cultures are incubated with the sense L, and the sensor fluorescence is used to rank the clones. Importantly, the fluorescence readout correlates well with D-lactate titers measured by HPLC, especially at medium to high concentrations, showing that sense L enables high throughput screening of D-lactate producers using a simple plate-based assay. In their third application, authors used the sense strain to detect the PETA's activity through TCA assimilation. Biological PET upcycling became realistic with the discovery of Ideonella sakaiensis, which degrades PET into intermediates like BHET, MHET, TPA, and ethylene glycol. However, screening new PETases, kinases, and engineered strain is still low throughput and time-consuming. In this book, the authors ask whether whole cell biosensors can accelerate such screening. As a first step, they engineered the Pseudomonas putida sense to express the TPH operand from Pseudomonas umsonensis, enabling growth on the PET hydrolysis product TPA with biomass tightly correlated to MSGFP fluorescence. To speed up TPA-based screening, the authors evolved the Pseudomonas putida sense T on TPA, doubling its growth rate within 1 week and reaching 0.64 per hour after 8 weeks from initial 0.28 per hour. This evolved strain is called sense TE. Sequencing showed general adaptation mutations in the chromosome and a key A to G mutation near the ICLR-RBS on the TPA pathway plasmid, predicted to increase ICLR and TPH expression. Reintroducing this single mutation into the original plasmid reproduced the fast growth and shorter lag on TPA. Sense TE then showed a linear fluorescence response to TPA, with about 80 for the signal change, clearly outperforming reported transcription factor-based TPA biosensors and motivating its use for PET degradation assays. The authors next used their TPA sensor strain to build a more sensitive PET degradation assay. They start from the evolved TPA sensor, Pseudomonas putida sense TE, and add a codon-optimized Ideonella sakaiensis MHET8, including its native secretion signal, by mini-TM5 insertion selecting the fastest MHET growing clone as sense TM. To also consume ethylene glycol, e.g., they inactivate the GCLR repressor by CRISPR base editing, depressing the GCL operand and enabling EG assimilation, yielding the fully engineered strain sense P. SenseP now grows and increases fluorescence in response to MHET and EG. Unlike SenseTE, they then use SenseP and SenseTE to read out PET hydrolysis by the chitinase LCC under different temperatures, times, and enzyme concentrations. SenseP gives about 3-fold higher GFP than SenseTE, and its readout correlates well with HPLC measurements of TPA and MHET, with enough sensitivity to detect lower than 50 micromole products, supporting high throughput PTAs condition and variant screening. In this experiment, the authors used the SenseP strain as a living reporter for PET breakdown in co-culture. They engineered a PET-degrading Pseudomonas putida PTD that secretes PTAs via a purine F signal peptide, and confirmed PTA's activity by BHET plate halos. In liquid co-culture, PTD grows on glucose, while SenseP can only grow on PET degradation products. So any SenseP growth and MSF GFP signal directly reflects in vivo PET depolymerization. With BHET and PETD present, co-cultures show similar growth of both strains, but about 22-fold fluorescence increase after 80 hours compared with controls, with signal rising from about 18 hour onward as PTD first generates substrate for SenseP. Overall, the figure demonstrates that this whole surveillance approach can quantitatively report plastic degradation by engineered bacteria in real time. In conclusion, the authors present a generalizable whole cell biosensor strategy in Pseudomonas putida based on synthetic orthotrophies complemented by target metabolites and readout via OD and fluorescence. This growth-coupled platform can detect diverse metabolites in monoculture, supernatant, and co-culture using only plate reader assays. The system is modular and tunable, by changing which central carbon pathway is disrupted and which metabolite restores growth. New sensor strains can be built with minimal extra engineering. Compared with conventional transcription factor-based biosensors, this approach avoids the need to evolve or discover specific regulators for each new analyte and integrates easily into high-throughput DBTL workflows. The authors also highlighted limitations. It cannot sense biologically inert or non-assimilable molecules. Complex mixtures with many similar compounds can reduce specificity, and co-culture sensing may produce metabolism by consuming the product. This is the end of my presentation. Thank you for your listening.